last page. So we are on 8.4, and 8.4 is primarily, totally about enzymes. So we'll probably spend a little bit of time making sure we're comfortable with what enzymes are and what they do. So we'll start there, even though I'll go this. I'll probably do some work to make sure we're capable and we understand exactly what an enzyme does in order to actually talk about how it's made and all the particulars about it that come from these questions. So the number one thing, you know how we've said certain things. We said, water is polar. Behavior is a response to a stimulus. So we want to be sure that another one of our just statements we always make, if there's anything about an enzyme, we want to be sure that the people who are reading our work know that we know enzymes are proteins. So, that being the case, I'll stop the video just to remind you. I'll go through this first, but I'll remind you of tertiary, secondary, primary structure, all those things, because enzymes are going to work because of the shape they have. So, what we've learned is there's this connection always between structure and function. So real quickly, since enzymes are proteins, they're going to have levels of structure. So we're going to go back over those right here. And I hope they mean more to you now that you made your insulin molecule. So insulin is not an enzyme, it is a protein. So we're going to be sure we clarify that level of structure of all proteins, of which enzymes are one type. When we have these, and they're just long strips. So the chain A was, I think, 21 amino acids long, and chain B was 30 amino acids long. Each one of those parts would be called a polypeptide because they have more than one peptide bond, and those would be your primary structure. So you can write it this way. This is what it is. It's primary. So for a protein, knowing we're talking about all proteins here, they have a primary structure which is just the order of the amino acids. So you might have, which we did do. Hold on, let me go find my strip. So this is the polypeptide we made. Not as fancy as this one, but still pretty awesome. If you didn't make it, or you can't find it, I told you we would keep coming back to it. This would be a time to pause this and make another one, because it will be one of the things I check on the notebook check, which will be coming up soon, because we'll finish chapters 7 and chapters 8 right before Thanksgiving. So we'll take the test the week before Thanksgiving. I think it's the week of the 16th, but I haven't looked at the calendar, but it's something like that. So, again, if you haven't made this, let's make it, and I'm going to walk you through what it is. I'm just going to put it back in my little section for proteins when I come back, but I'm going to use it here. So I'm going to take this strip and I'm going to remind myself of what these are. So in this particular case, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 amino acids. The primary structure would be that I would read these: glycine, valine, histamine, serine, tyrosine, all of those. That would be my primary structure. So it's the order of the amino acids, and they're going to be, all of the amino acids are going to be held in place because of the peptide bonds that are between two amino acids. So held in place by the peptide bonds. Now, just a little fact, when you think about it, your number of peptide bonds is always going to be, if you think about it, I'm going to have a bond between 1 and 2, 2 and 3, 3 and 4, 4 and 5, 5 and 6, 6 and 7, 7 and 8, 8 and 9, 9 and 10, but there's no bond over here. So there's going to be one less than the number of amino acids. So it's going to be the number of amino acids you have minus 1, because you don't have a dangling bond out here at the end. So that's the primary structure. And because we wrote the properties of these amino acids, we understand that that's really important. But it was much more evident here. So these dark sections, these runs of dark color, what we learned was the properties of those amino acids cause them to have associations with each other. So this primary structure determines all the rest of the structure. So we'll put that here. So it determines, because of the placement of where they are, all of the higher secondary, tertiary, quaternary levels of structure. That order of the amino acids. Okay, so that's primary. Secondary, this is when we learned, whoa, I know what I want to do, but my hands don't want to help me do what I want to do. She's telling me to wrap this stuff and tape it and hold it all at the same time. You begin to see how structure leads to function and how difficult maintaining this structure could be. So the first thing we did is we worked with the green and we held amino acid 1 and we created these turns, these helices, through the association we had between the amino acids 1 through 9. Those were able to form alpha helices. That's secondary structure. So secondary, now that you have all of your amino acids in the order that they are, the close proximity that they have to each other, they begin to have associations with each other, and there's two types of shapes that you're going to get as a result. So for secondary, there's going to be hydrogen bonds that are going to form between amino acids. And that's what we showed here. So there's a hydrogen bond between amino acid 1 and its carboxyl group, because there's a carboxyl end of the amino acid, and there's an amine side. Just reminding ourselves here for amino acids. Let's come down here. You're going to have a central carbon. You're going to have a carboxyl side. You're going to have an amine side. Then you'll have a third carbon, a third hydrogen at that third spot for the carbon. And then this is the part that changes to make the difference between a glycine, a valine, a histamine. This is what gives the properties. So this is where the functional groups go. The functional groups give the amino acid its function. All the rest of this stays the same. So you're going to have hydrogen bonds that are going to form. So here's what we formed. Between amino acid 1 and 5, there's a hydrogen bond between the carboxyl end and the amine end of these two different amino acids. So they have this attraction and they have a hydrogen bond. That would be like the tape. Of course, proteins don't have tape. So they're going to have an association, a bond, a hydrogen bond, weak by itself, strong when there's a bunch of them, and also another one that 5 is going to go with its carboxyl side and the amine side of amino acid 9. That's going to form an alpha helix. So there's 2 primary, secondary structures. I won't say primary, that's a bad way of wording that. There are 2 secondary structures. One would be an alpha helix, which we have 1, 2, 3. Those were the curls we made. The other that hydrogen bonds form, so the order of the primary sequence, when they're next to each other, they begin to have attraction through hydrogen bonding. That's where you get these secondary structures. The other ones are beta sheets. That's where we had that run with the arrow, right here with 24, 25, and 26, and it has this pleated or condensed, if you think about it, structure. So they're going to be tighter and they're going to have this accordion-like sheet pleat structure that's also held by hydrogen bonds and is secondary. Tertiary is where you start having these functional groups interact with each other. So in our paper here, this is where we said, hey, these are nonpolar, hydrophobic, charged or polar, hydrophilic. So when they have a positive or a negative or a positive, that's charged. That's going to associate well with water. Polar, great with other polar things like water. Nonpolar, those will not associate well with water. Hydrophobic, they're going to be attracted to each other and then repelled by water. So these residues, these amino acids, their R groups are nonpolar. They're going to associate with others that they can get close to, and they'll dive to the middle, which that was when we started doing some of these weird things. So we made turns. Remember the fold that we made? There's one turn. That now creates a 3-dimensional structure. We made these 2 turns here, and then we started having disulfide bridges that were between the cysteine at, well, that's about to tear off, 19, and then of the blue and 20 here. So those sulfhydryl groups that are that, functional groups main property giving, then they come together and you end up with 2 of the sulfur groups together, and then that was when you had to do the weird twisting. So that weird twisting is what took us into this being in the 3-dimensional shape that it is. That's tertiary. So this is where you get your 3D shape, and it's going to be from the properties of the functional groups: hydrophilic, hydrophobic, nonpolar, same thing as hydrophobic, charged, polar, same thing as hydrophilic. So of the functional groups at the R position, those were the ones that varied of the amino acids. So down here, all of these groups, that's what gives this amino acid its characteristics, and that's how we know which one it is. It can go from being something simple as one methyl group to a whole bunch of things, including rings, down through here. So that tertiary structure, including the disulfide bridges, that all of what I kept calling our foot, all of these, the orange, the green, the yellow, are associations that are tertiary because of the functional group that's sulfhydryl, and you get other ones that have that other types of shape giving properties from functional groups, but that just happens to be this insulin's main folding So this is going to give you that whole 3D shape. So when you have more than one polypeptide chain together, which we had chain A and chain B, so you have tertiary because there's more than one polypeptide. So I said that wrong. Quaternary. Say it again, say it right. When you have more than one polypeptide chain, you have quaternary structure that comes from taking the 3D folded up one polypeptide, another 3D, it could be 4. There's a lot of tetramers that are out there, hemoglobin, that have 4 exact polypeptide chains put together that give this quaternary shape. So this is going to be where you have more than one polypeptide. The only reason I wanted to go through that again is because we haven't gone through it since we folded. So I hope you see how this works, because we're going to come back to this with, if I didn't have tape, that would be a really different kind of situation. Like, I don't know if I could keep the coils coiled. I know for sure it'd be like precarious. I wouldn't want to have the conditions change very much. And that's what real proteins live in, is it has to be just right for the shape to stay the way it is. So I'm going to put this little guy back here where the proteins are. And now I'm going to talk about this active site. So knowing that we've got this 3D shape, this 3D shape is going to determine structure, determines function, how this actual protein can work. And for enzymes, their 3D shape is primarily focused on the active site. So I'm finally going to actually start with number 1. So your enzyme, that's a protein, enzymes have this 3D shape, and it has a pocket to hold a particular molecule called the substrate. So if you think about the enzyme as being the worker, the substrate is what it works on. So enzymes have a 3D shape that has an active site. Let's talk about what the active site is. So the active site is a location. It's like a parking space for just one type of molecule. And that's based on, you can see that if you've got something that's this structure, not everything's going to be able to move and dock. So because of the shape of the protein, in this case an enzyme, it's going to have a unique active site that is available there for a molecule to dock. So the active site is the place, the parking space, where the substrate, what is being acted upon, the molecule that where'd that go? Substrate will dock. Okay, so here's what I want to do, because we're going to continue to add things. I'm not going to do these in order. I'm going to do 1, and then 4, and then 5, and then 6, and come back to 2 and 3. So we're at 1, we have an enzyme, and what we're going to do is we're going to work on building a more complete model of what an enzyme looks like. So the more information we have, we'll come back to our figure, our model, and we'll update the model. So going on what I know now, here's what I know. I know that I have a molecule that has a three-dimensional shape. So it's hard to do this. Think how hard it would be to draw this, which I have, which we're going to include in our notebook. We have that. See how that there's 1, 2, 3 of them together? So we're going to be able to see that it is really difficult to draw something that's three-dimensional in 2 dimensions. So we're going to give ourselves a little bit of freedom to be simplified here. So what I'm going to do is I'm going to make my enzyme a simple shape, knowing that that's not the case in real life, but for drawing's sake. And I'm going to give myself, so here's the enzyme, and I'm going to make a uniquely shaped active site. So let's make something like this. So this is the enzyme, which is a protein. The shape it is based on its primary structure of the amino acids in a row, its secondary structure of all of these alpha helices and these beta sheets, its tertiary structure where we had to twist and turn all of that until it folded on itself and we had 3D shape. That's what this is. So here's my enzyme that's a protein. This spot is the active site. It's the location in the enzyme that will be where the work is being done. It is a uniquely shaped spot because there's just going to be one molecule that fits there. So I'm going to have a molecule. Let's see if I can draw it over here. This will be my substrate, and it fits perfectly in the active site. So this is called, a lot of times they'll call this E, they'll call this S, and I'm now going to draw the ES complex, which means I'm going to redraw this piece. So here's E, and I'm going to dock the substrate. This is the molecule that is being, or molecules, that is being acted upon by the enzyme. It's what the enzyme works on. So let's go with an example because we have one from the lab we've done. Catalase is the enzyme that acts on hydrogen peroxide, so it's the substrate. So for our example, I'm just going to dock this substrate. So this is S, and when we see it in this together conformation, it's called the ES complex. So if there's another molecule, let's just draw something else. Get a different color. So maybe there's another molecule that looks like this. It will not fit there. It's just going to be this substrate that has the active site exactly made to fit it. So, now that we've gotten ourselves built up to the shape of it, how does having that particular shape that can hold one particular molecule lower E sub A, well, you have to know what this is. This is the energy of activation. So now we're going to step out of this whole process one more time, and we're going to build the case for what an enzyme is used for. Okay, so we're going to take a break for just a second, and we're going to talk about just energetics in a simple form. If you think about it like this, some reactions will naturally occur. Processes will naturally occur. They're favorable conditions for that to go from reactant to product. Some reactions going from reactant to product, the conditions are not favorable or likely that they would happen on their own. These are going to be reactions that would happen naturally, that are going to occur as long as the conditions are met to where that this process can occur. It looks super simple. So, okay, we've got this ball. Of course it's going to roll down the hill. That's not the case. That's not going to be the way things work. With this kind of simplified energetics, there would be little control. Reactions would not be able to be either paused and then we let them happen, or where that they would all just proceed. This one would not proceed, this one just would. So what happens is this. When you have reactions that have, let's say, two molecules that are going to be brought together. So we'll make this is molecule 1, and we're going to bind it to molecule 2. This process is called dehydration synthesis. We're going to form a bond there. And when we're done, we'll end up with a product that looks like this. I think we can see what that is. Here's the issue with it. In order for these to occur, like falling downhill, both of these reactants have to come together at the exactly right level of energy and orientation, the way in which they're turned. So the way we've drawn this orientation would be the only way they would work. I always make this as my example. I'll use my glue stick again. Is there going to be the ability for this cap to go on the glue stick? Yes. This can occur. This fits. It's made for it. This could occur. What's the likelihood that the cap will be oriented in a way that it will go on the glue stick? Here's what we're looking at the difference between. And forgive me if this is loud, because I have no idea if it's going to be. If I set these both down here, and I just let them, whoops, see what I'm talking about? Let them just move randomly. Yes, there might be, at some point, the orientation that's correct, but that activation energy, that energy of the actual orientation, the environment, everything being just right for that reaction to occur, that's what all of this is. This is what an enzyme changes and lowers. So instead of this random hope you get together, you've got an enzyme. If you think of my hands as one enzyme, there's going to be in the active site a perfect place for this glue stick to sit and this cap. And so what would happen naturally, as long as they were in the right orientation, is now there's no wasted energy or needed energy for them to get to this place. I've taken all that out and I've put them in the spot where this should occur. That's how enzymes lower activation energy. Reactions that would happen anyway over a really, really slow, long period of time, enzymes speed it up by, here you go, do your thing. because they will bring together the components in the proper position, orientation, environment that they need to be in. So once these are with proximity to each other, the reaction will occur. Now this product isn't shaped the same anymore, and it will dissociate, and the enzyme's ready to do it again, because the active site is created specifically to hold this glue stick in this position, the glue stick cap in this position, and both of them in proximity in the right orientation. Not this way, not that way, not this way, but in the proper orientation of the two, so what would naturally happen happens, and then the enzyme is not used up, just like my hands aren't. So there are also reactions where that you're breaking a bond, and what enzymes do is they stress or torque that bond so that while it's quite comfortable like this and stable, it's not stable anymore when I have put stress on that bond, to the point that it will break and dissociate, and now that reaction has occurred. Those reactions, we know, have a different name. I'm so sorry. So if I had this molecule, and we're going to break them apart, which is hydrolysis, that would be the equivalent of that torquing of that bond to where that these two pieces now cannot stay together anymore under that pressure, and you end up with a broken molecule into its pieces, its products. So this would be the reactant, those would be the products. So all the enzyme is doing is providing the proper place with orientation for these two processes to occur. So I also want to put, while I'm here, I want to just remind you of what are some other words that could be used for these. So as we are dehydration synthesis, we're making a bond. So let's come over here, and I'm going to put what other words could be used. Making bonds is called anabolism. So it's anabolic. So you are making bonds. Whenever you see bonds, it's going to require bond energy. So you're going to have an energy source. So this requires energy. Often it's paid for by what we talked about before through using our currency of ATP. So it requires energy, and we're going to call this process also endergonic. So all of these words, along with dehydration synthesis, describe this same process of putting things together. When you break something apart, that's called catabolic. Cats break things. Just to help you remember the difference. You're going to break bonds. You're going to release that bond energy. Which we call that exergonic. So these processes are going to be coupled together these endergonic and exergonic processes, we can couple these together where that the energy that's released from here is going to go into ATP and be used to fuel these processes that require energy. So this is that energy coupling we were talking about. Now that we understand about what the whole enzyme piece does, let's write that over here. So enzymes, their job, first of all, they're proteins. They work by reducing all of that random proper orientation, proper energy of the collision, energy of activation. So enzymes work by reducing energy of activation, which was the whole point of this question, which is E sub A, and if you don't, you know it's right here. And here's how they do that. This is for reactions that would normally occur. They don't do magic. Enzymes aren't magic. You can't speed up something that would never happen anyway. So these are for reactions that would normally occur, but just way too slow for life. You know that whole meme of it, ain't nobody got time for that. So nobody's got time for all that. Hello, are you orienting correctly? We're getting to it. We're trying. We are. Nobody has time for that. So enzymes are going to work by, yes, the reaction would normally occur ridiculously slowly. So what are they going to do? They reduce the energy of activation by providing, so they're going to provide the substrate or substrates with the proper, the best, the optimal positioning orientation so that the reaction can occur. So I've got you right here, do you think? Okay, you're going to occur anyway as long as you can get lined up in the proper position with the right amount of energy. I did that for you. Have at it. And they will. And so this is all because this proper positioning is that active site. It's about structure. Because of how I'm made, guess what? I'm going to hold you. You just like this, and you just like that. I'm an enzyme. You're going to naturally then, because you're in the perfect position, the reaction's like, yep, rolling downhill. Easy peasy. And we want to be sure that we recognize that enzymes are not used up in the process. That didn't even make sense. My hands are absolutely still here, ready to receive another glue stick and cap until, that I myself, as a protein, lose my shape, which means I can't work, or I am tagged to be broken down because there's no need for me anymore. Okay, so we've answered 1. The active site is the location where the perfect position is given to the reactant or the reactants so they can do their thing and have the reaction they would normally have just too slowly. We're going to come back to 2. We're going to come back to 3 because we're going to now draw in another piece of the enzyme. They're a little more complicated than my little circle, and we're going to add in these other pieces. So the next piece we add in is a cofactor, and then we're going to look at different ways that we can, when we say inhibit, people feel like that's a negative connotation. And I get that. Inhibit, though, just means control. Think about it this way. If you did everything that you ever thought about, wow. Thank goodness for natural inhibitions. That ability to control. That's what this is. Enzymes, when they're made, you would not want to have a machine that was on all the time. Think about how weird that would be. So this morning I got up and I rolled my hair. So my rollers would always have to be on. I ate a piece of toast, so my oven is now still on. I washed my hands, so my faucet would have to still be on. I opened my door, so it would have to stay open. Let's say I'm a crazy person and I mowed my yard before I got here, so my lawnmower is still out running in the yard. And I drove a car here, so my car is now running in the parking lot. No, we make things, but we naturally need to inhibit them from being all the time doing their thing. That's all this means. And then we'll talk about specificity based on, we said that there's this unique fit. There's descriptions about how that model is. Okay. Where were we? We were adding a cofactor to our picture. So, there's really 2 pieces to this. What number is that? 4 to 4. So enzymes, most enzymes, don't work alone. So I don't know how it is for you, but if you think about levels of control, instead of being frustrated, if I told you, hey, your group, you have 3 people, we cannot start the work until all 3 of you are here. If one person rarely shows up, we don't do the work, right? They've controlled whether the work can happen or not by their presence or absence. So enzymes having buddies, parts, is a level of control. So you could make all of it and it would still not work because the one piece you need isn't there yet. That 3rd group member hadn't shown up. So enzymes have other parts that go with it. One is called a cofactor. The other is called coenzymes. Most enzymes have cofactors. So I don't have any vitamins with me, but I know you've seen. Have you ever wondered, like, who the heck needs manganese or selenium? Why would you put that in there? Well, right here's the reason. Magnesium, zinc, manganese, selenium, all of those are cofactors that are part of the enzyme. So a cofactor, obviously when I said manganese, you knew I wasn't talking about a protein, right? They're non-proteins. They are often metals, like what I talked about. So they're often metals. That's why these are in your multivitamin. And you might have things like zinc, you might have iron, magnesium. Those are super common. And what they do is they're often part of the active site. So often part of the active site. And because of their properties, they have this stabilizing effect on the whole enzyme because of being in the active site. So they stabilize the active site, which just means allow it to stay in the shape it's in an easier, less energetic state. Like it's not as hard for it to hold that shape. Now you know why everybody talks about zinc with COVID. Because whatever you're doing to fight COVID would involve zinc, and you would need to make many, many, many of the molecules that have zinc in them to stabilize the shape. That's what it is. So, I wanted to stop and go find something, but I think I'll start over here. So let's just talk about what we were talking about before. Sometimes, most of the time, an enzyme has built into it a cofactor. So inside the active site will be some metal. That reason we were saying, who needs manganese anyway? Well, those kind of weird things that you find in your vitamins and minerals, they're there because if you don't have these, the enzyme will not work. It won't be able to function. And so they'll have things like zinc fingers, they'll have different names that they call them, but these are required. They're cofactors. They're not just non-proteins. To be more specific, they're inorganic, which means they have no carbon in them. Well, that makes sense because zinc is zinc, right? Now, there's also a partner with enzymes called coenzymes. These are also not proteins, but they are organic. They're really usually very small. They're molecules that are required for this enzyme substrate complex, this putting together this assembly to actually work. And I bet you've heard of some of them. So let's see if I can find why I stopped this, because I was trying to think of them in my brain. Sorry, my computer's doing a lot of things and it's really slow. Come on. Let's let this minimize. Okay, maybe you won't. Let's go here. Let's go here. And here's a list. So, did you know why you took vitamins? Right. Did you just take them because somebody said they're good for you? Well, what do? They are modified to become coenzymes. There's two types. Prosthetic group. Do you know what that word means? Prosthetic. So, if you have seen someone who has been, I don't know, like a veteran, and they've come back and they're missing a limb, they have a prosthesis, right? So they have this added on piece that's necessary. That's what a prosthetic group is. So it's going to add on to the shape, and so that's what this does. A co-substrate is going to have the, hey, I know you're going to go in that active site and you don't want to go in there by yourself. I'll go in there with you. And it goes in so the substrate is able to fit into that active site. So there's two different types. We don't really talk about them. But, so, a lot of these are super interesting. So vitamin B3 is niacin. And it's going to form through some chemical reaction changes. It becomes NAD. Holy cow, is that going to be an important molecule here soon when we start doing photosynthesis and cellular respiration? Because it's an electron carrier. Let's see what else we've got here. Coenzyme A, that's going to be part of also what we do when we talk about cellular respiration. For acetyl-CoA, you may not remember any of these, but all of these vitamins in this form are not going to be a coenzyme until they undergo change, and then they become a coenzyme. Let's go back to what coenzymes are. add those in there. They're these little organic molecules, but they're not a protein. They're required for this ES complex to actually occur. And we learned, and what I just said was, sometimes it's, hey, I'll go with you, substrate, let's go in that active site, come on, let's go together. Or there'll be an added-on prosthetic part to the enzyme. But all of them are required for this to work. So, you may see some words like apoenzyme, which is the enzyme without the coenzyme. When you have the coenzyme added, you get something, and let me write it correctly, holoenzyme, which is the actual functional ready-to-go one. So examples here like coenzyme A, NAD+, which is from niacin. So these pieces we're now going to add on to our enzyme model. So I think what I'm going to do is I'm going to color code. So I'm going to make my substrate green, and I'm going to make my enzyme brown, pick any color as you want, and I'm going to make my cofactor pink, and another color. I'm going to make my coenzyme orange. So I'm going to draw that now, updated. So this was what we understood to begin with. A little too simplistic, right? Hey, an enzyme that properly holds its only unique substrate in this active site. We've learned that there's more to that. So let's update that. So I think I can, I'm not sure, I'm going to try to do it right here. So I have all of this in front of me. Excuse me, I hate that happens every time. So the cofactor is going to need to go and sit somewhere in this active site. The coenzyme is going to either be prosthetic or an added-on piece to the enzyme, which is what I think I'm going to draw, or it's going to be in the active site with the substrate as a co-substrate. But I think I'm going to just draw the prosthetic version. So I'm going to draw my enzyme. So here it is. Why is this so hard for me to draw in my brain? So there's my active site, and we said the cofactor needs to be in the active site, right? So this is my pink. So what I think I'm going to do is I'm going to make this piece right here, this sitting inside of the enzyme, that's going to be my cofactor. I know that because it's pink, right? And I can draw here that this is the cofactor, stabilizing that active site. Then for the enzyme, it's going to need to have a coenzyme with it, which I decided I was going to do is some kind of part that's added on. And I'm going to include another place on this enzyme, another location. So when we come back and I do the next part of this video, then it's going to be where we already know where it is. So this is the active site. This is where the substrate goes. And I love the definition of what I'm about to put. It's a site that's not the active site. So if I called you and I said, hey, listen, where are you? I'm at a place that's not my house. What a terrible way to tell me where you are. But that's the way that they define this location. So we're going to put another place on the enzyme. This is called the allosteric site. It is a site that is not the active site. So I'm going to keep going with my enzyme. I'll go ahead, color it in. And then I need to put that prosthetic piece that's required, that's a coenzyme. So the complex now for the enzyme would be the protein enzyme, the metal, often metal, cofactor that sits in the active site and helps stabilize this active site, the organic but not protein small piece that's the coenzyme that's either a prosthetic group added on to the enzyme or sits in the active site with this substrate here. We didn't draw that one, but it's a coenzyme, which just means me too, right? Not a protein, small little molecule. Then I want to draw my substrate, which would go in here, which would have this shape. I went ahead and just gave it some crazy things to show it's not something simple. So this is the substrate that would sit in this active site. Right here is the active site. And this is the substrate, the molecule that the enzyme is going to act on. Okay? So I think what I'm going to do is stop this video, and I'm going to do a whole separate video on 2 and 3, 5 and 6, so that we can break it up. How's that sound? Okay, let's do that.